So hi everybody, this is Julian speaking. I hope you can hear me well. We just started the webcast. I will wait for at least another minute to allow everybody to join the webinar since the number of attendees is still growing. So let's wait another minute and then we'll get started. Okay. So welcome everybody to this webinar. I'm very happy to guide you through this presentation. It's part of our Selling Webinar series. And this week we will talk about setting up efficient cell line development workflows. My name is Julian. I'm the CSO of CYTENA, and with me I also have Fabian, our CMO, who will help me with the Q&A session later. So thank you very much for joining. The presentation will last around 35 minutes, and then we will have plenty of time to answer questions. Many of you might use the GoToWebinar plugin for the first time. So let me briefly introduce you how this works. You should see this control panel here on your screen. The arrow allows you to access further options and also to maximize your window. You can actually ask questions throughout the whole presentation. We will then address them after the slides, and you can do this by typing them into this questions text box that you see here. We also uploaded some material that you can download, for example, an app note or on the F side, and also a brochure for our devices. At the beginning, I want to briefly introduce who we are and what kind of products we are developing. Since last year, we're actually part of a larger corporation, the Selling Company, and Selling has been founded in 2016 and has grown rapidly since. And by now, we are present in over 60 countries and have offices all around the globe to develop new products and support our customers. This drive for innovation enables us to significantly grow our product offering, which covers a number of different areas, most prominently bioprinting, but also liquid handling and single cell isolation, single cell analysis, and imaging. Here at CYTENA, we develop instruments for automated isolation of single living cells since 2014. All our instruments are capable of label-free cell isolation and come with image-based assurance of clonality. It started all off with a single cell printer that you can see here that was launched in 2015. In 2018, we then launched the Xcyte series, which comprises 3 instruments for isolation of mammalian and microbial cells. And lately, we also started to ship the Seabird, a device that enables early suspension culture in 96 well plates. Most of the world's top pharma companies are CYTENA customers. And it's really exciting to see that there's a lot of very prominent companies in the pharma space, but also in academia that use our instruments to develop their cell lines. So what can you expect from today's webinar? Let me briefly go through the agenda. After introducing the most prominent applications that are addressed by our customers, I will explain how our single cell dispensing technology works. Then I will discuss some important aspects of single cell cloning workflows for cell line development. And then I'm very excited to share one of our most recent developments in this webinar, the Seabird, a novel well plate-based microbioreactor for early suspension culture. Let me provide a quick overview on the most prominent applications that we focus on. Our instruments are already very established in cell line development. There's a lot of little details and also variations to this workflow, but from our perspective, it typically starts with genetic engineering or transfection of a mammalian host cell line. Then followed by single cell isolation. And I believe that many of you are aware that assuring clonality in CLD workflows is an important step to meet the requirements of the FDA and other regulatory bodies. Well-growing clones have to be then selected for productivity, have to screen for quality, and also the stability of the cell lines has to be assessed in order to finally choose the production clone that produces the therapeutic at the end. Another application we are focusing on is cultivation from mixed microbial samples. Typically, this starts with a heavily heterogeneous sample. And the third application is single cell analysis, and most prominently single cell genome and transcriptome sequencing. Today, we will focus on cell line development, and there will be 2 more webinars next week and in 2 weeks on the other 2 application areas. So what are the major challenges in cell line development? Having worked now for several years with some of the leading CLD groups, it boils down to 4 major challenges. And the first challenge is assurance of clonality, which is also a regulatory requirement. The second one is clone recovery. You want to make sure that you recover a decent number of clones, well-growing clones from your cloning process in order to be able to select and upscale. And downstream of the single cell cloning, the major challenges are associated with cell line stability and productivity, as well as product quality. These are all parameters that have to be guaranteed for the final production clones. The current gold standard for assuring clonality is automated well imaging. This technology, however, is not error-free. Looking at the literature, you see a lot of in-depth studies of different imaging technologies, and it can be frequently seen that 1 to 5% of the cells that are deposited into well plates are not detected by the imager due to artifacts and edge effects. And this can lead to so-called ghost wells, wells where no cell is detected, but growing colonies emerge. And today I want to talk about 2 technologies that address some of these challenges associated with CLD workflows. Most importantly, the assurance of clonality and also clone recovery. And I want to introduce you to the single cell dispensing technology, which allows deterministic cell isolation with image-based assurance of clonality, and which also results in a high cell viability. And the second application I will talk about is a plate-based microbioreactor for early suspension culture, which allows to better reflect the final culture conditions present in the bioreactor already directly after single cell cloning. Here you see one of our devices, the Seaside. It's our instrument for label-free isolation of mammalian cells. It's a benchtop instrument that comes with a software that is super easy to use. And if we zoom into the printhead here, you can see the nozzle of our disposable dispensing cartridges. We use an optical detection system similar to a bright field microscope to detect the cells in the nozzle of our dispensing cartridges. And only those droplets that contain a cell with the right properties are then deposited to the target substrate. This is one of the cartridges, of the disposable cartridges that we use. And now I would like to show you a short video that shows the instrument in action. So this video has shows you how actually the typical workflow looks like, which start with filling a cartridge with a cell suspension, attaching it to the instrument, and then setting up your workflow in the software, and then you're able to dispense individual cells into the well plate. And after a few minutes, you will end up with a well plate full of single cells in each well. So let me go back to the slides. And let me go a bit more into detail and explain how the principle works. So this slide shows a schematic of our nozzle of the dispenser. Here you see the nozzle with some cells inside. We have a tiny vacuum suction which is placed directly underneath the nozzle and our target substrate. And we're continuously monitoring the nozzle area, and using automated image processing, we count the number of cells and determine their morphology. And if there's no cells present in the nozzle, we have the pneumatic shutter switched on, and the droplet will be removed directly after ejection. If there's a single cell present in the nozzle with the right properties, the pneumatic shutter will be switched off and the droplet can be dispensed to the target substrate. And of course, if there's multiple cells, the droplets will be also removed. In the Xsite series, we have 3 instruments. We have already seen the C-side in the video. We also have an instrument that comes with an additional fluorescent detection channel that allows you to also sort and isolate cells based on fluorescent intensity, the F-side, and an instrument with a high resolution optical detection system for isolation of microbial cells such as bacteria or yeast, the B-side. All our instruments use sterilized disposable cartridges to prevent cross-contamination, which come in packs of 10 and are individually bagged. And since this is the only part that gets into contact with the sample, there are no routine cleaning procedures required. And it also eliminates the risk of cross-contamination between samples. For each cell that is dispensed, a series of images is stored to the hard drive of the instrument that document how the cell was isolated. Let me now show you another video that illustrates how the software looks like. So here you can see how the software looks like. You will select a plate format. In this case, it's a 96 well plate. Then you can set up your experiment by looking at the scatter plot. You can determine the right parameters that you're gating for. Here we set the size and the roundness of the cells. And we can also determine the number of cells that we want to isolate. So in this case, we have a few control wells, for example, well number A1, where we dispense 5 cells, and in all the other wells, we will dispense a single cell. We then press play. The software lets us confirm that we set up everything properly, and then the isolation process starts. And in the upper right corner, you see a live video stream of the nozzle, of the dispensing. You see the cells approaching the nozzle and how the algorithm detects them and dispenses the droplets with the cells. The progress can be monitored by looking at the plate. Each time the check mark occurs, a single droplet with a single cell was dispensed into the respective well. For 96 well plates, this takes typically between 2 and 5 minutes. So let me go back to the slides, and I want to show you another example of our nozzle images to illustrate how it looks like on the F side. Here you see a single GFP transfected CHO cell some way above the nozzle, and our laser is actually switched on. In order to prevent the dye from premature bleaching, we only illuminate the area around the nozzle with the laser. So far we don't see a fluorescent signal. When we now continue, the cell gets closer to the nozzle, reaches the nozzle area, and here it is detected. We now see the fluorescent signal. This is an overlay image of the bright field and the fluorescent channel, which becomes a little bit more clearer if we zoom in. We then have another image which shows the image processing overlay, and the final image which shows the nozzle after the single cell has been dispensed. And this illustrates that the cell has actually left the nozzles. And many of our customers in the pharma industry include these nozzle images to prove clonality of their production cell lines to the FDA or other regulatory bodies. One exciting application of the F-side that I want to highlight and share with you today is a workflow for cloning CRISPR-Cas9 edited stem cells. Our partner at the University of Freiburg used our instrument to isolate this rather delicate cells after transfection and genome editing. And the F-side helped a lot to simplify this workflow, especially since only small sample volumes were available after transfection. And as you can see, with up to 40% clone recovery in their plates, they reached a very decent outgrow, given that these are very, very delicate cells. When it comes to what can be achieved with respect to assuring clonality with our technology, I'd like to refer to this publication by Mandy and David from Genentech. They actually established a fully automated workflow with the single cell printer. In their validation round, with over 3,000 clones, they achieved a single cell isolation efficiency of 96%. And the paper provides a detailed description on how thoroughly they assessed the probability of clonality. They demonstrated that using a state-of-the-art plate imager in combination with the SCP, with the single cell printer, that this results in a probability of clonality that exceeds 99.99% and therefore clearly meets the common requirements by the regulatory bodies. They also state that the SCP results in dramatically reduced number of plates needed for cloning compared to other automation workflows. If you're interested in learning more about assurance of clonality, the pros and cons of plate imaging and appropriate metrics, I can really recommend these two publications. A bunch of our customers are amongst the authors, such as, for example, the group at Amgen. Please shoot us an email if you're interested in getting the original publications. We might help you here. So now I want to take the time and pass two questions to the audience. The first one is, what is your biggest challenge you're facing in your CLD workflow? So you should now see a poll popping up. And I would like to ask you to, take a few seconds and think about your challenges if you're working in this field or if you have been in touch with people in this field, and just vote for this. So I will wait another 20 seconds and then we will proceed to the next question. If you are facing other challenges that are not listed here, please also feel free to comment on this by using the questions text box. We are happy to discuss this later in the Q&A session. All right, let me now go to the second question of today's webinar. The second question will be about the number of clones that are needed for a typical project. So I believe a typical project, most people are looking for a few hundred of clones, but this can really vary a lot depending on the product and also the transfection method, obviously. So I'm super curious to see what is your take on this. How many clones do you typically expect from your single cell cloning workflow that needs to be recovered for upscaling, selection, and product quality assessment? So I will wait another 10 seconds and then let us go back to the slides so we can proceed the presentation. It's really great to see that a lot of people are participating in the poll. So let us now go back to the presentation. In the second part of the talk, I will now introduce you to the Seabird system, which is one of our latest developments. It's a system that brings suspension culture to the very beginning of the CLD process, and it can be rapidly integrated into common CLD workflows. So this slide shows the system. This is a special lid that can be placed onto standard 96 or 24 well plates, and this is a control box with a docking station, but we will talk more about this in a bit. So on this slide, you see a typical cell line development workflow as it is performed by many of our pharma customers. After single cell cloning, the cells are cultured under static conditions for several weeks before well-growing clones are then transferred to shaken deep well or 24 well plates. And after additional cultivation, selected clones are then transferred to larger volumes and finally to shake flasks or mini bioreactors such as the Amber 15. And one of the major issues here is that during the first weeks after cloning, cells are cultured under static conditions which do not match the culture environment the cells experience in later stages of the process and during production. It is well known that this can lead to instabilities. Further, once a critical cell number has been reached, oxygen supply can be limited. And the CBIRD system was developed to bring cells into suspension culture and mimic the culture environment of later stages from the beginning on in the CLD workflow. Here's a slide that illustrates the principle of the CBIRD. As I mentioned, it's compatible with standard well plates. It comes with a special lid that you can see here, and the CBIRD control box, which houses a pump and the electronics. The lid contains 96 little tips that gently move the liquid in the wells up and down, and the tips are designed such the cells are continuously brought into suspension. I will now show you some data from experiments that we conducted on CHO cultures with and without the CBIRD system. In this case, we used a suspension cell line, a CHO-K1 cell line expressing a therapeutic protein. We did this by monitoring, by pipetting cells into well plates, into each well of a well plate, and then monitoring the cell density, the protein yield, and the volume-specific productivity per cell in static cultures and in CBIRD suspension cultures. And here you see the results from two individual experiments. One has been conducted in Germany and the other one in Asia. And in both cases, the use of the CBIRD lid results in significantly increased cell numbers after 3 or 5 days. The relative titer increased more than twofold in both studies, and since a higher protein yield can be expected from a higher number of cells, we also calculated the relative volume-specific productivity per cell per day, which you can see on the rightmost plot here on the third graph. And as shown here, cells grown as suspension are also more productive. This slide shows the average cell density of in one of these experiments over the course of 5 days, clearly illustrating that cells grow faster and a higher cell density can be reached in the CBIRD culture. And we also developed a second version of the CBIRD system that's compatible with 24 well plates. And using this system, we again see much higher cell growth with the CBIRD lid, enabling the suspension culture. Why is this the case? This data here demonstrates that one of the main reasons for this is limited oxygen supply in static culture conditions, which is basically the gold standard, right? Cultivation in well plates. So here we actually use sensors at the bottom of the wells on the well plates to monitor the dissolved oxygen during cell culture. And under static conditions, this curve here, the oxygen level drops to less than 10% within 3 hours after we started the culture. While the CBIRD, with the CBIRD lid, the oxygen level drops significantly slower and stabilizes at much higher levels, close to 50%. In gray, you can actually see the oxygen level of a control experiment. This was a well plate with pure medium, and obviously here the oxygen level stays constant. So this is our CBIRD system. It comes with a docking station and 3 control boxes that allows you to process up to 3 96 well plates or 24 plates in parallel. The system is quite compact and fully autonomous. It can be placed in an ordinary incubator so it's compatible with standard lab equipment. And we think that the results from this and other studies are super exciting. We believe that the CBIRD system can increase the efficiency in upscaling suspension cell lines after single cell cloning, and thereby shorten the time you need to find and select the high producers easily by 1 to 2 weeks. So if you're interested in adopting this technology, please come and talk to us. We are currently running early access program for the CBIRD. Now I'm almost at the end of today's presentation. So let me briefly summarize the key features of our Xsight instruments for single cell cloning in CLD. I've shown you already a video of the C side, and we had also a look on the nozzle images that are generated by the F side. In the next webinar, we will talk about the B side. All these instruments come with image-based assurance of clonality. They are benchtop instruments, and the software is designed in a way that they are easy to set up and easy to use. They all use the single-use cartridges, which eliminate any risk for cross-contamination, and you can expect single-cell efficiencies that exceed 95%. They also guarantee high cell viability, and the throughput, I mentioned this already, is 2 to 5 minutes per 96 well plates, or around 10 minutes for 384 well plates. And we also have an automation API, which allows you to remote control the software from an automation server. So if you're interested in integrating our instruments in an automation pipeline using plate handlers and so on, please come and talk to us. We can help you to set this up. So how can we come together? We are happy to schedule individual telecons or web sessions. We then typically proceed with an on-site demo in your lab to introduce you to the instrument and allow you to work with your cells and study efficiency and growth. I assume that most of the audience is still affected by the COVID-19 implications, and due to severe restrictions in travels, we have actually set up a live demo lab here in Germany to offer online demonstrations on request. So if you're interested, now is the chance to get an application-specific live demo that you can watch from anywhere. So please get in contact with us, and our team is really happy and ready to prepare a demo for you. Now we are at the end of today's presentation. This was the first event of this Selling CYTENA webinar series. Next week, we will talk about the isolation and cultivation of single microbial cells from mixed heterogeneous samples, and also anaerobic cultivation. And in 2 weeks, we will host a webinar on single-cell genomics workflows. So if this is of interest for you or for your colleagues, please let them know and sign up. With that, I'd like to thank you for joining. I hope you enjoyed the presentation, and I'm now ready to take your questions. So give me a few seconds. We will have a look on the questions and go through them. And we are also happy to address any further questions after the webinar via emails or phone calls. So let me have a look. So the first question I see here, can you share the CRISPR related paper? So yeah, there is actually a paper which is currently prepared. As far as I know, it's not published yet, but I believe we can share already some of the data. So I think a good idea would be if you get in touch with us, and then we can send you some data and also help you to address your application if you're interested in sorting CRISPR-edited stem cells or CRISPR-edited cells. I believe there will be also a paper later this year published in a scientific journal. Second question is, can I use a green and the red fluorescent label with the F-site? In general, the F-site comes with a single detection channel, and but it also allows you, as all our other instruments, to detect and isolate and sort the cells based on bright field images. So so you can sort based on fluorescent intensity, and we are happy to also integrate a red laser if you're interested in a different dye. But in general, it comes with a single fluorescent channel and allows to sort based on morphology and also based on fluorescent intensity. Another question is, how do you make sure that cells are sticking together, are not dispensed? I guess, so if I understand this question correctly, it's about how we ensure that we do not dispense multiple cells in case they're clunking or sticking together. That's a good question. We are, as I already mentioned, we are using bright field images where we assess the morphology of our cells and other objects in the nozzle that will be dispensed. And two cells that are sticking together have a very different morphology compared to a single cell, a single suspension cell, for example, which is typically a round cell. So this can be very nicely addressed by selecting 4 rather round cells, and we see that if we choose these parameters appropriately, then cells that stick together are put to waste. This can be also seen very nicely in our nozzle images. How long does it take to dispense into the plates, and which formats are possible? That's also a good question. So all our instruments support SBS plate format, and by default we are able to dispense into 96 well plates or 384 well plates. We can also use PCR well plates, so not only culture plates. And for 96 well plates, it takes between 2 to 5 minutes, and for 384 well plate, it takes approximately 4 times longer, a little less, around 10 minutes for a typical experiment. And it's also possible to use other formats. So if you have a specific question for a specific format, please get in touch with us. But in general, our software can be adapted to also dispense onto other formats as long as they fit the SBS footprint and are flat substrates. So there's a question regarding the seabird. How often does the seabird stir? How much liquid does it stir? So this is something you can actually set up. The docking station can be connected to a computer, to a laptop with USB, and this allows you to set up your mixing protocol. And we have some default protocols where we know that they work well with, for example, a standard 96 well plates filled with culture medium, but this allows you to also change the settings, increase the volume or decrease it. But typically you can imagine that it should be around 50% of the volume that you pre-dispense in your well plate to make sure that your wells are not sucked dry. Also the mixing period, you know, how often will be mixed can be set up, so you can actually control this. So there's another question regarding the plates, if there's limitations, for example, height or color. In general, there's no limitations. Typical standard plates can be used. Of course, at some point, the height might be a limit, but we haven't really seen any standard well plates or standard PCR well plates that do not fit into our instruments. For the Seabird, the Seabird is actually designed for standard 96 culture plates. We recommend Eppendorf plates, but the system can also be used with other plate formats. We're happy to look into this and help you choosing the right plate. But to begin with, we would recommend Eppendorf 96 well plates, and we can help you choosing the right plate here. There's another question regarding the Seabird. Can you please tell us how the cell numbers were calculated for static and suspension cultures? So in this case, we actually took samples of each well and counted several times the number of cells in these samples with an automated cell counter to get these numbers over the course of several days. And then there's a follow-up question, how the relative productivity was calculated based on the cell number and on titer measurements that we did. We could then calculate the productivity per cell per day. What are the conditions that are to be considered for an incubator while using the Seabird? So in general, the system really is designed that you can put it in any standard incubator. It's quite compact. The idea is that you really can use the settings and the environment for your cell culture that you're used to or that you set up. So it's compatible with standard incubators and for mammalian cell culture. I think I now went through most of the questions. So if there's more questions, and I think there's a bunch of questions which are bit more complicated, we're happy to address them afterwards via email. So please get in touch with us. We're happy to help you, happy to set up a live demo. Again, thanks a lot for joining the webinar today, and have a great day.